Je me présente, je suis le professeur Patrick Bogui de l'INRS ETE. Donc, j'agis aujourd'hui à titre de représentant du comité de programme pour la soutenance de thèse de Madame Indrani. Alors, permettez-moi dans un premier temps de vous présenter les membres du jury. Donc, nous avons le professeur Peter Jones de l'Université de Sherbrooke, qui agira à titre d'examinateur externe et président du jury. Nous avons le professeur Gérald Zaguri, professeur donc à l'École polytechnique de Montréal. Il agit à titre d'examinateur externe. Le professeur Jean-François Blair, qui est aussi directeur du centre, qui agit à titre d'examinateur interne. Et le professeur Rajesh Tyagi, professeur donc à l'INRS ETE, qui agit à titre de directeur de recherche de la candidate. Alors, le déroulement de la soutenance se fera comme suit. Dans un premier temps, nous allons donc entendre la candidate pendant 30 à 40 minutes. Elle va nous présenter le fruit de ses travaux de recherche. Ensuite, nous aurons à observer 5 minutes de pause pour permettre à ceux qui veulent partir de partir. Et suivons la période de questions avec les membres du jury. Après quoi, nous allons donc demander au public de poser éventuellement des questions. Et sur ce, nous allons donc nous retirer avec les membres du jury pour délibérer. Donc, avant de céder la parole à la candidate, je vous demanderai de fermer vos cellulaires pour éviter de distraire la candidate et d'éviter de vous abstenir de prendre également des photos. Et nous allons donc, la porte restera fermée tout au long de la présentation de la candidate. Madame Indrani Bhattacharya, vous avez déposé une thèse intitulée « Inhibition de croissance de Candida Bruxelles et évaluation du profil nutritionnel de la fermentation de Saccharomyces unispora à partir de lactosérum. Le jury l'a jugé acceptable et soutenable. Vous avez maintenant 30 à 40 minutes pour nous en faire la présentation.
Good morning, everyone. Good morning, jury members. Mon projet, inhibition de la croissance de la Candida bruxelles et évaluation du profil nutritionnel de la fermentation de Saccharomyces unisporus et partie du lactosérum. Je suis présenté mon présentation en anglais. To start with, I will be explaining the introduction, the problem areas that I dealt with, methodology, hypothesis, objective, originality, result discussion, conclusion, and my contribution and recommendations. To begin with, what is lactoserum? Lactoserum comes from milk through pasteurization, either through acidification or enzymatically, and we get cheese and lactoserum, which has high concentration of fats, which is being skimmed, pasteurized and concentrated, and then we get the concentrated lactoserum, which through further ultrafiltration we get retentate and permeate. Right now the global scenario of cheese whey production worldwide is 0.19 million tons. Cheese whey can be characterized physically and chemically. In physical characterization, I will be explaining the protein and fat which is high in concentration in milk, and there is slight fat in lactoserum and no protein casing in milk. In chemical characterization, there are 3 different types of cheese whey: acid whey, sweet whey and salt whey. Salt whey has high concentration of lactose and protein in comparison to sweet whey and acid whey. Currently, cheese whey is utilized worldwide for value addition in forms of value-added products and single-cell protein. In form of value-added products as bioplastic, enzyme, EPS, organic acids, and for the production of single-cell protein, usually the contributors are yeast, algae, fungi, bacteria. The major yeast contributors for single-cell protein are Candida species, Clubberomyces species, as bacteria species like Lactobacillus, Bacillus licheniformis. The context of my project is why the production of single cell protein, two major organisms, K. marxianus and L. fermentum, would have been the contributors unless an unknown pathogen, Candida krusei, which invaded into the system, and the final product was only Candida krusei and K. marxianus. The temperature was maintained at 40 degrees Celsius at pH 3.5, and there were no traces of Saccharomyces unisporus. According to the choice of organisms, the acceptable organism is Saccharomyces unisporus. In North America, though it is not accepted under Canadian Food Inspection Agency and FDA, however, it is accepted in the European Union by International Dairy Federation and European Food and Feed Cultures Association. However, Candida krusei is totally unacceptable throughout the world. So the problem areas are for the production of single cell proteins, for food and feed production, any form of biological or chemical contamination has to be eradicated out, and organism, microbe which can associate with single cell production can be inserted into the medium. So my major problems were Candida krusei was coming as a contaminant for the production of single cell protein. For the production of single cell protein, culture, mixed culture, SCP producers would be a better biomass than the monoculture. Right now, currently the monoculture is K. marxianus. In developing countries, 80% of the people are lactose intolerant, and apart from that, cheese whey leads to higher COD when disposed of into the environment. So my hypothesis is, hypothesis 1, can Candida krusei be eradicated out from the system either chemically, biologically, or through bio-nanoparticle approaches? Hypothesis 2, can Saccharomyces unisporus be a better SCP contributor and can be grown as a mixed culture along with K. marxianus? My hypothesis 3, can acid hydrolysis of cheese whey will be proved economic in comparison to enzymatic hydrolysis? So my principal objective is elimination of the contaminant Candida krusei during the fermentation of the mixed culture and addition of Saccharomyces unisporus for single cell protein production. My specific objectives were evaluation of the chemical and the biochemical approaches, which is going to be only Candida krusei and without having any effect on K. marxianus or Saccharomyces unisporus. My objective 2, which is the production and optimization of the bio-nano conjugate, which is going to have an effect on Candida krusei and not the acceptable yeast, marxianus and unisporus. Objective 3 is the study of the nutritional profile of unisporus. Objective 4 is hydrolysis of cheese whey for the production of mixed culture S. unisporus and K. marxianus. Originality of my work is inhibition of Candida krusei with the help of a killer protein, conjugation with a metal ion. The killer protein comes from W. saturnus. I would like to explain a little bit about W. saturnus. It is an organism acceptable by all the regulatory bodies, and it is from the dairy industry. My second originality is nutritional profile of S. unisporus because it has never been studied before, and originality 3 is the utilization of culture mixture of K. marxianus and S. unisporus for single cell protein production. Methodology 1, my objective 1, production and optimization of the bioconjugate for the single cell protein production, where Biochemistry saturnus was grown in cheese whey, molasses and urea. Biochemistry saturnus is completely lactose. It does not consume lactose and it consumes sucrose. That is why molasses was added. However, cheese whey has high content of certain major minerals, which will be a nutrient broth for Biochemistry saturnus, and carbon and urea is the nitrogen source. Further is the optimization of the procedure, where for the nanobioconjugate formation, different concentration of silver nitrate and different volumes were considered, along with different volumes of W. saturnus supernatant per 5 ml of water, distilled water, at different temperature at various time period. At every 2 hours, samples were collected and it was verified using UV vis spectrophotometry. Further, the characterization of the conjugate was carried by centrifugation, filtration using 0.45 micron filter, and characterization using FTIR, zeta sizer, SEM and EDS. Further, the conjugate was tested in weld diffusion method. My objective 2 were the evaluation of chemical, biochemical and nanoparticle approaches were tested against inhibition of Candida cruzi, where macroorganisms considered were Candida cruzi, K. marxianus, S. unisporus and W. saturnus. Lyophilization of the supernatant of W. saturnus and lyophilization of the entire organism, and then study of the inhibition. Inhibition studies are based on chemical, biochemical and nanobioconjugate methods, where chemical approaches were different temperatures ranging from 20 to 45 degree, pH from 2 to 9, various different H2O2 conditions with as minimum as 100 ppm and as maximum as 4000 ppm at different temperatures. NaCl concentration from 1.5 to 2 molar were chosen. The biochemical approaches were chloro oil at various concentration and lyophilized powder of W. saturnus at various concentration, and the synergistic effect of H2O2 with the lyophilized powder of W. saturnus were tested. The nanobioconjugate is the conjugate which we got from objective 1 at various concentration. It was tested against the mixed culture. My objective 3 is nutritional profile study of Saccharomyces unisporus, where the substrate utilization profile was tested, were tried using biolog system, and the different growth parameters were studied, which is variation in carbon source, variation in nitrogen source and variation in pH, where the different carbon sources chosen were cheese whey, molasses, glycerol. Different nitrogen sources chosen were urea, ammonium sulfate, and at various pH from 3.5 to 5.5. And finally, the samples were taken for statistical analysis using principal component analysis technique. My fourth objective is hydrolysis of cheese whey and permeate to provide nutrients for Saccharomyces unisporus, where hydrolysis of cheese whey and permeate were carried out with H2SO4 at various ranges with temperature treatment of 121 degrees Celsius at different time period with as maximum as 120 minutes. Then analytical methods used were analysis of carbohydrates, analysis of volatile compounds, analysis of amino acids, analysis of ethanol, metals and total protein. So my result and discussions, starting with objective 1, where a protein profile study of W. saturnus shows that the maximum protein concentration was observed at 12 hours and further down the line at 21 hours. However, cheese whey has an initial protein concentration of 4.5 gram per liter. So the total protein concentration of W. saturnus, the initial protein concentration of cheese whey has to be deducted out. It is a visual study where at 0th hour there was no bioconjugate formation, so it was a transparent solution, and at 12 hour formation of lightish yellowish brownish color gives the formation of the nanoparticle. Further, optimization of the concentration and volume of silver nitrate along with different volumes of the supernatant, where it was observed that at 0.1 molar of AgNO3 we have the highest number of bioconjugate cells. The highest SPR peak demonstrates the number of cells. In this case, the peak has to be in between 300 to 500 nanometers with the highest peak. Highest peak resembles the maximum number of cells. When 200 macroliter of 0.1 molar AgNO3 was taken against the maximum number of bioconjugate cells were observed at 200 macroliter per 5 ml of water. The supernatant concentration required was 180 macroliter of supernatant per 5 ml of water. total protein study was conducted, which is required for the synthesis of the bioconjugate. However, the process started just at 0th hour, but it continued till 12th hour. The objective was to get maximum cells of the bioconjugate. A time period study was observed where it is seen that when we are taking 180 microliter of the supernatant, we are having the bioconjugate with as low time of 4 hours, but when we are decreasing the supernatant concentration, we are getting it at 12 hours. Further, the characterization shows that the biomolecule is getting attached, the biomolecule which is responsible for reducing silver is having a crystalline structure. Since we filtered it using 0.45 micron, we are getting a 200 nm size for all the particles, and EDS study was confirmed just to verify that there is no metallic contamination and just silver is the only metal present in the system. Further, an FTIR analysis shows if there is a variation in between 900 to 1900 wave number, the contribution is from the amides. Amide 1, 2 and 3. Amides are from protein all the time. So if there is a variation in amide 1, that is definitely from the core of the protein, that is the alpha helix. If there is a variation in amide 2, that is from the beta sheet, that is the secondary part of the protein. And finally, the tertiary structure of the protein, where there is a contribution of amide 3, and no other biological particle is contributing for reducing silver, and just the protein is the contributor. Further, the bioconjugate is tested against Candida krusei, K. marxianus and S. unisporus, where it was observed that through a well diffusion method that the activity of the protein, which otherwise would have been as low as this picture, it has gone 3 times with the nanobioconjugate, and then we went for scale up. I would like to describe the resultant description of objective 2, where first approach was chemical approach with NaCl and chloride tested against the mixed culture of Candida krusei and marxianus, where it was observed that though Candida krusei is getting eliminated, but the concentration is too high to carry on the experiment with. Therefore, we eradicated the chemical approach and we went for biochemical approach, where H2O2 with as low as 300 ppm can inhibit Candida krusei in a monoculture, but not in a mixed culture, where Candida krusei does not have a catalase enzyme which K. marxianus has. Once H2O2, once it goes in the intracellular region of Candida krusei, the hydroxyl ions, it goes and attack to the mitochondria, which is the powerhouse of the cell, and which further damages the DNA. The experiment was conducted at pH 6.0 and temperature 28 degrees Celsius, but in a mixed culture and with a lesser pH. There was no inhibition. However, there was slight decrease in the cells of Candida krusei. Further, the temperature was raised from 28 to 40 and then further 45, and then the addition of W. satinus, which has the capacity to produce killer protein and can kill Candida krusei, were added into the system to have a synergistic effect, and it was observed that H2O2 is diminishing from the system because W. satinus even has catalase activity similar to that of K. marxianus. So Candida krusei is no way killed. Then further increasing the concentration of H2O2, it was observed that at a very high concentration of H2O2, it is killing Candida krusei with a slight decrease in K. marxianus. So we restricted ourselves to W. satinus, where the concentration of the killer protein were tested against the total protein, and it was observed that if 156 microgram per ml of killer protein is there in the system, is going to kill Candida krusei. Therefore, the lyophilized supernatant of the protein of W. satinus was tested again 2400 ppm H2O2, and it was observed that at 12 hours there is no Candida krusei, but the concentration of H2O2 and the lyophilized powder is too high to work on with. Therefore, we moved our focus to the conjugate, and there it was observed that at the bulk preparation, the time period which we earlier demonstrated as 12 hours has ranged from 12 hours to 48 hours. to have the maximum number of cells of bioconjugate. Therefore, the conjugate was tested against the mixed culture where 350 micromolar of the conjugate can kill Candida krusei just after 0 hour. And it has slight effect on K. marxianus. Because silver is basically highly antimicrobial and killer protein usually has no effect on K. marxianus. The stability test of the conjugate was conducted for 20 weeks and it was observed that in the mixed culture, Candida krusei is not coming up and K. marxianus is retaining the CFU pyramid throughout for 6 months. My objective 3, which is the nutritional profile study of Saccharomyces unisporus, where Saccharomyces unisporus is a non-lactose consuming organism, non-lactose assimilating organism too. Other carbon sources chosen as sucrose and glycerol, though glycerol is though Saccharomyces unisporus is partially consuming glycerol. The molasses at 7%, which has 10 gram per liter of sucrose along with urea 0.22% weight by volume. Saccharomyces unisporus is having a growth profile of 10 to the power 8 at 30 hours. And LCMS analysis shows that there is complete consumption of sucrose till 42 hours. However, since it's a non-lactose consuming organism, lactose stays in the system. Utilization of glycerol shows that it is hardly, Saccharomyces unisporus is not consuming glycerol at all, so we did not go ahead with the glycerol as a carbon source. So considering molasses 7%, we changed the nitrogen source from urea to ammonium sulfate of 0.4% weight by volume and again the growth profile. The exponential phase was achieved at 10 to the power 8. Variation in pH showed that at pH 5.5 it is having a good growth profile similar with that of 4.5 and a lower growth profile at 3.5 with all the major minerals like calcium, potassium, sodium, sulfur, magnesium. When doing the nutritional profile of a yeast, we have to consider all the aspects of the yeast that the yeast will be producing as an intermediate, of which one of them is fatty acid, and it was observed that in saturated fatty acid, palmitic acid is the Saccharomyces unisporus is producing maximum of palmitic acid with a with a comparatively high concentration of polyunsaturated fatty acid and slight low of monounsaturated fatty acid. The total fatty acid concentration is 0.86% weight by weight, and according to FAO result in single cell protein biomass, the total fatty acid concentration should be less than 3% weight by weight. For the amino acid analysis against total protein, it was observed that Saccharomyces unisporus is producing 4.2 gram per liter of total protein, where the essential amino acids, methionine, it is barely producing any methionine, and 3 amino acids like histidine, phenylalanine, and tryptophan. Because there is a less production of histidine, phenylalanine, and tryptophan because of metabolic restrictions. Because other essential amino acids other than histidine, phenylalanine, and tryptophan, they are produced just after glycolysis in the TCA cycle, whereas histidine, tryptophan, and histidine, phenylalanine, and tryptophan are produced in the second phase, that is the non-oxidative phosphorylation, non-oxidative pentose phosphate pathway, which are they produce aromatic amino acids, and these three are aromatic amino acids. My objective 4, where hydrolysis of cheese whey and permeate were conducted at 0.28% H2SO4, where there was no hydrolysis of cheese whey. However, there was hydrolysis of permeate because of stoichiometry disbalance. There was, though hydrolysis took place, no equal moles of glucose and galactose were formed because of free loss of glucose and galactose. Further hydrolysis of cheese whey with a raised concentration of H2SO4 gives a hydrolysis of cheese whey at 100 minutes, but again there is stoichiometric loss. At 0.4 weight percentage of H2SO4, where there is stoichiometric balance of glucose and galactose when converting from lactose. This graph is before the hydrolysis of cheese whey, where the lactose is intact, and this is after the hydrolysis, where it is broken down into glucose and galactose. Saccharomyces unisporus was grown as a monoculture and then as a mixed culture along with K. marxianus. Unisporus was not growing at all in cheese whey or permeate because of nutritional restrictions. But after the hydrolysis, it started growing by consuming glucose. But it is not consuming galactose. However, in mixed culture, K. marxianus is consuming the leftover lactose, glucose and galactose, which is a restriction for Saccharomyces unisporus. The amino acid profile of the mixed culture was considered where there is an increase in arginine, asparagine, serine, alanine and glycine. Another study conducted was qualitative analysis of the intermediates. So out of all the intermediates we derived, certain intermediates could be of biological importance if considered on separate aspects. However, no qualitative study on it has been done. So my conclusions are: approaches for killing Candida krusei, either chemically or biochemically, are expensive, not economical. And the conjugate with 350 micromolar containing 1 ppm of silver at pH 5.5 and 30 degrees Celsius can kill Candida krusei in a mixed culture of KM and Candida krusei. This conjugate is a stable compound because it is not forming any metallocomplex with the minerals present in cheese whey. The same inhibitor can eliminate other biological contamination because of the presence of silver. S. unisporus is strictly urea negative. The monoculture of Saccharomyces unisporus has high concentration of lysine, glycine, asparagine and valine. It has less protein. Protein concentration is 4.2 gram per liter with fatty acid concentration of 0.86% weight by weight, and it is 80 rich when compared with Saccharomyces cerevisiae, which is the model yeast. The mixed culture produce higher concentration of serine, alanine, glycine, asparagine and alanine. My contribution towards science and technology with aspects of fundamental science and applied science will be: in today's world there is high utilization of antibiotics and antimicrobials ranging from 60,000 tons for 2010, and in 2030 it is expected to reach 100,000 tons. More use of antimicrobials or antibiotics is actually causing a resistance in the pathogens. So the biological inhibitor, which can be either protein, like in my case it is protein, it is never going to cause that kind of resistance to any pathogen it is going to target. For example, Candida krusei. The approach is green. It is economic if we are doing in a lab scale, and it can counteract antimicrobials for killing specific pathogens. My second half of the study, which is Saccharomyces unisporus, though one part of the world it is accepted and other part of the world it is not accepted because of the reason that maybe the nutritional profile was never been done before. So after the study, it has the same traits of that of a grass, which is generally regarded as safe organism, similar to that of K. marxianus. It has all the traits of producing single cell protein, similarly to that of K. marxianus and Saccharomyces cerevisiae. My recommendations would be, since the killer protein of double Saturninus was never been filtered out and there was total protein conjugation with the metal ion had taken place if the particular protein is being filtered out, and then the which is being after and then conjugated with the metal ions, I'm sure the concentration from 1 ppm of silver will go down to less than 1 ppm. In the second half of the study, the recommended ideas will be study of the intercellular and extracellular proteins, enzymes. If unisporous is taken for food and feed production, removal of nucleic acid has to be there, because 2 gram of nucleic acid present in single cell protein is considered as toxic, and quantitative analysis of the intermediates has to be. I would like to thank Professor Tyagi for allowing me to work under him and allowing me to work on various horizons of biotechnology. I'm thankful to Professor Blake, Professor Vijaypuri, and Professor Jones for reviewing my thesis. I'm thankful to Dr. Bejawada, Dr. Chandran and Dr. Yan for helping me with my research work. I'm thankful to my colleagues for helping me throughout. I'm thankful to INRS for providing the excellent facility for all sort of experimentations being conducted here. I'm thankful to Professor Valero for helping me with research. I'm thankful to my friends, my family, and God Almighty. I'm thankful to everyone for coming.
Merci à Madame Indrani pour sa présentation. Nous observons 5 minutes de pause pour permettre à ceux qui veulent quitter de bien vouloir le faire dans la discrétion.